Good morning students, welcome back to our today's video session, that is of chapter number 8, reproduction. Okay, so before we move into our today's video session, let us discuss what we have learned in our previous video sessions. So in our previous video session we have learned about the next two methods of vegetative propagation. One is budding, another one is spore formation. In the budding process we have seen that the new organism has developed from the body surface of the parent plant in the form of a small outgrowth, which we are calling it as bud, okay, which developed from the surface of the parent body due to repeated cell division, that is in the hydra on a specific site. Due to repeated cell division, a small outgrowth is developed, which we call it as bud. That bud slowly grows in the size as an individual, and finally after attaining a specific growth, okay, it gets separated from the parent plant and develops into an individual or as a new organism. Next is spore formation. This we people are noticing in the lower simple multicellular organisms like rhizopus, one of the fungi. And here what is happening over here? The rhizopus are developing the new individuals or the new ones by the formation of spores or seed-like structures. So these spores or seed-like structures which are developed which are developed by the rhizopus, they all are developed through a bulb or blob-like structure which is present at the surface of the sporangia, known as sporangiophore. In this they are developing sporangiospores, which later on burst open to spread into the surrounding environment. Under favorable conditions, they give rise to the new rhizopus plants. Now, in this today's video session, we will be discussing the next mode of reproduction, that is our vegetative propagation. Now, what is vegetative propagation? Here in this vegetative propagation, okay, the plants are using their vegetative parts like roots, stem or leaves. The plant will be using their vegetative parts to propagate or to generate the new individuals or the new offsprings. So I can say that vegetative propagation can be defined as a mode of reproduction where vegetative parts like roots, stem and leaves are used by the plants to develop the new individuals or the new plants. Okay. Now there are different ways of performing vegetative propagation. Okay, what are those different ways? One is natural method, another one is artificial method. Let us see what is happening in the natural methods. Look here, natural method in the sense here is there is no involvement of human beings. There is no involvement of human beings. Therefore, it is considered as natural method. So here, in this natural method of vegetative propagation, vegetative parts of the plant, either it can be root, either it can be stem or either it can be leaf. Any part of the plant will be using to develop a small outgrowth or a small swelling as from the bud region, known as vegetative propagule, which after attaining certain growth detach from the parent body and can be developed as a new plant. Okay, that means I mean to say from the nodal region of the plant or at the bud region of the plant, a small outgrowth will start developing either from the root nodules or from the stem or from the leaves, which we call it as vegetative propagule. And this vegetative propagule, after attaining certain growth, gets separated from the parent plant and develops into a new individual. This we call it as natural method of vegetative propagation. Okay, here the plants which are propagating with the help of roots, examples we have like Dahlia, asparagus. These two plants, they use their roots to develop the new individuals. And here they also certain plants which develop with the help of stem like potatoes, ginger, garlic. Okay, these are the plants which are propagating by using their stem. And there are certain plants which propagate with the help of leaves, that is by bryophyllum and begonia. So if we just look out at the leaf of the bryophyllum plant, Okay, it will have notches at the leaf margins. So under favorable conditions with the availability of soil and water, these notches start developing roots and leaves from the nodes of the leaf or at the notches of the leaf. Okay, and they can be separated and can be grown as a new bryophyllum plant. So in this way they will be using their leaves to develop the new individuals or the new plants to propagate. That is what is happening in the natural methods. Then we have artificial methods. What is actually happening in the artificial methods? Here in these artificial methods, these are actually used by the plant growers and horticulturists. Plant growers and horticulturists, they use this artificial method of vegetative propagation. Okay, that means we human beings are involved in this one. This is man-made methods where we people are developing the new plants. Okay, so here we people will be using again the vegetative parts of the plant to develop the new plants from them. So it is also one of the asexual mode of reproduction, but as we are involved in this one, we refer it as artificial method of vegetative propagation. This we people perform through three different ways, that is by the cutting, layering and by the grafting method. First of all, what is happening in the cutting method? In the cutting method, like it is carried out in the rose plant. Okay, so in the rose plant, the healthy part of the stem or the healthy branch is selected of the rose plant, which doesn't have the capability to generate fruits and seeds. So here these plants we people are propagating with the help of their vegetative part like stem. So a healthy stem or a branch part of the rose plant will be selected and a slant cut, again I am saying a slant cut is made at the nodal region. So at the nodal region a slant cut is made, and this cutted part will be incorporated or will be kept in the auxin for few days so that it can develop the new roots. Okay, and later on it will be transferred into the soil where now it can receive the minerals and water with the help of roots from the soil and can develop as a independent individual or as an independent plant. That is in the cutting method. Then in the layering method, what is happening in the layering method? Here also in the layering method, we people will be selecting any healthy branch of the jasmine. Jasmine plants specifically they are propagated by the help of layering. So a specifically healthy branch of the jasmine plant is selected, and here a half moon shaped cut, I am repeating, on the branch a half moon cup shaped cut is marked at the nodal region. But here we people are not separating that part of the branch from the parent plant. It is still attached to the parent plant, whereas in the cutting we have separated it from the parent plant, but in the layering we are not separating it from the parent plant. Just a half moon shaped cut or half cup shaped cut is made at the nodal region, and now it is bent into the soil and a heavy object is placed on its surface so that it doesn't pull out of the soil. So after a few days, as it is continuously receiving the nutrients from the parent plant and soil, because under favorable conditions this nodal region will develop the roots in that region. Now it gets fixed into the soil with the help of roots and has got the capability that it can absorb the nutrients from the soil with the help of its roots. Now we can separate it out from the parent plant and it can be independently grown as a new jasmine plant. That is in the layering method. Then the third is grafting method. What is happening in the grafting method? This grafting method is one of the advanced method or mode of reproduction carried out by the artificial method in the vegetative propagation. So here we people will be selecting two plants. One is considered as the stalk, the base of that one will be considered as stalk, and the hybrid part which we have to incorporate, or which the desired characters which we want to incorporate the surface of that plant is known as scion. Okay, so here this stalk is the part of the plant, or the part of the wild plant which has got the capability of disease resistant or surviving all the extreme climatic conditions. The base part of that one we people are selecting, and the surface of desired quality of the plant, that means better quality fruit production, okay, but it doesn't have disease resistance, so that part of the plant will be selecting the surface one, that is the scion, and it will be incorporated into the stalk. That means the cambium of the two plants is joined here by tying them very tightly, so that they can fused, the cambium of both the plants fuses together and unites together, okay, and can be grown as a single independent plant. In this way we can obtain the desired plant what we want, okay, according to our wish. And this is basically performed in the fruit plants like mango, guava, apple, okay, so that we can obtain these hybrid plants which can survive all the extreme climatic conditions and also produces and gives rise to a better quality food production, fruit production. That is in the grafting. That is about the grafting. Okay, the next is tissue culture. This is an advanced method of vegetative propagation. It means this tissue culture technique is basically performed in vitro conditions, okay, where we can produce the similar varieties of plants on a large scale, that is on a large number, in a very short period of time. What is happening in this tissue culture technique? In this tissue culture technique, we people will be selecting the group of cells from the actively dividing cells present at the tip of the stem or at the root of the plant. That means from any of the nodal region or at the tip of the plant, we people will be selecting a group of cells. That group of cells will be transferred into a test tube in the lab, where we people have created the medium, okay, no, or we have generated or we have prepared the medium with all the requirements which is required by the plants in natural climatic conditions. So all those requirements are available to the cell, from where it can obtain the nutrients, water and sufficient optimum temperature with the heat and light. Now this cells will be transferred into many test tubes, single cell in every test tube, and they will be kept with all the requirements. Now after certain days, these cells, which are actively dividing cells, they goes on with the continuous cell division and develops into a group of cells which we call it as callus. That group of cells we refer as callus. Now this callus, which an undifferentiated group of cells, later on undergoes differentiation process and develops roots, leaves, okay, from this group of cells. That means later on grows into a new individual or into a new plant, which now can be transferred into natural climatic conditions or into the soil, where they can be grown independently. So here in this tissue culture technique, we can obtain the exact copy or the clone of the desired parental plants which we want in the future generations, so that we can grow all them together on a large piece of land, or they can be supplied to a number of people at the same time with similar characteristics. So horticulturists use this advanced technique, tissue culture technique, to develop the plants which have very particular desired qualities which they want to, okay, now supply to the people. So in this way, tissue culture technique we can use for developing such plants. So overall I can say that vegetative propagation has got many advantages. That means this vegetative propagation method we people can use to develop the new plants from such plants which doesn't have the capability to generate fruits, seeds and flowers. So those plants which even lost the capability to develop the fruits and seeds and flowers, they can also give rise to the new plants, or new plants can be obtained from those plants by using their vegetative parts. Secondarily, similar type of plants can be obtained on a large scale, on a large number, through this vegetative means of propagation. That is what is happening. That is what the advantages we people have got through this vegetative propagation methods. Okay, that's all students in this today's video session. In the next video session, we'll be discussing about the sexual mode of reproduction in the plants, how it happens and how it is carried out, and what the parts involved in the sexual mode of reproduction. Till then, have a good day, wonderful day, and enjoy your day. Goodbye, students.